Hold on. Right. So genetics, if you take a look, every year there's definitely one MCQ that is 1 out of 50, and there's a trend of asking 1/3 of a structured essay question as well, right? And always, every year almost, there has been a genetics short note, right? A genetics short note is becoming sort of a now a common thing, right? So if you take the genetics weight, right, definitely one MCQ, and it can be up to 2, right? Because from question number 40 to 50, a gap there can be additional genetics MCQ, right? In addition to the Animar MCQ, right? Compulsory 1 MCQ, and additional 1 MCQ can also be there. 1/3 of a structured essay is becoming the trend, and also a short note. Short note is becoming sort of like a regular habit, right? Almost repeating every year a genetic short note. Yeah, and there has been one example of an essay, right? Part of an essay in from genetics as well in a very recent past paper. So all the questions in the questions booklet are past papers, right? These are past paper questions from 2019 to 2025, right? I have included up to 2025 paper, right? That's why we have so many questions now, right? 6 years of questions. Yeah, and I have included maybe 1 or 2 whole syllabus 2018, 17 wage, 1 or 2 questions that you can do. I have included. So this is genetics, right? So we are going to go through the genetics concept very fast. Once we are done with the concept, we are going to do questions, right? That's the pattern. For example, when you are talking about test crosses, I'm going to give you test cross questions, so you can taggala methanama do it and get the relevant practice. Huh? So for genetics, I'm going to give you a small note as well, because the resource is sort of bit confusing and sometimes are කියවන්න ගියාම එපා වෙනවා resource එක. For genetics, if you want to quickly understand, because there's a lot of understanding in genetics, right? Resource එකෙන්ම understand කරන්න ගියොත් you are going to get tired of genetics. Genetics එපා වෙනවා, right? So in the, I'm going to give you a very small note that is going to include all the, let's say, like sort of a logical theoretical points for you to understand the concepts. But the main reference is the resource. දිගටම resource එක follow කරන්න ඕනේ. But the note is going to cover sort of the things that you can quickly refer. Taggala genetics මොනවා හරි අමතක වුණොත් go to the note and check, right? Okay. So I need you to have like a writing book, online people, right? A book to write down, and also the 2 tutes, right? The other tute contains only diagrams, and the second tute contains only questions, right? That's the plan. So those tutes are going to cover genetics and the next lesson molecular as well. So my plan is to finish genetics within 3 weeks. Genetics will be done in 3 weeks with all the past paper questions discussed as well, right? Within that time. Okay. So Taggala come to the resource book, page number 89, genetics, right? Genetics. So who is the father of genetics? It's Gregor Mendel. Taggala highlight, first paragraph. Father of modern genetics is Gregor Mendel, right? Mendel discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. So Pisum sativum, right? Pisum sativum is the plant he used, right? Garden peas. Based on that garden peas, he did a lot of experiments, right? Because you will see that his decision to choose garden peas is also a very scientific decision, right? හොඳට හිතලා බලලා තමයි garden peas choose කරේ. Okay, so we are going to see the importance of why he chose garden peas. It can be questioned, right? So His experiments were conducted decades before the concept of chromosome. So he didn't know anything about DNA. Mendel didn't know anything about DNA, how from parents to children, how characters pass. He knew somehow from parents to children, these characters came. He didn't know how. But he did experiments and he proved, yes, they are passed. He didn't know what is exactly the molecule. එහෙම DNA කියලා එකක් nucleus එකේ ඉන්නවා. There's a nucleus and there's a cell division, meiosis, right? Meiosis is going to break the two, uh, let's say, our paternal and maternal chromosomes into two, and how going to make a sperm and egg. And he didn't know those things, but he definitely knew from parents to children, these genes are coming, these characters are coming. So he had a lot of gaps in his understanding. He didn't know many things, but he was not bothered by those. He did his experiments and he published his findings, right? Later, with the new findings, we can fill in the blanks in his work, right? That's what happened. So, right, vocabulary in genetics, right? Vocabulary in genetics, that is very, very important. These definitions are very commonly questioned, right? So the first one, what you need to know is a character, right? So genetics is actually how we pass characters from one generation to generation, how we got our characters from our parents. So it's important to know, to start from the beginning, right? So important definitions, right? Write down, take your books, Puta, right? If you can write down these definitions itself right now is good, right? The first definition that you need to know is character, right? Study the definition right now. Dhamma පාඩම් කරගන්න definition එක, right? What is a character? A heritable feature. Write down. A heritable feature that varies among individuals of a population, such as hair color or eye color. ඒ ටික ඕනේ නැහැ, right? A heritable feature that varies among individuals of a population. That is a character. A heritable feature. A feature that you can pass down. That means, that is what you mean by heritable, no? What are other such characters? Skin tone, what else? Height, IQ, right? There are so many other characters that pass down from generation to generation, no? Then we come to sort of another definition that is actually coming under character, right? That is called the trait, right? So traits are actually something coming under characters, right? What is a trait? So if you take an example as your hair, let's say, what do you call, pattern of hair, or your type of hair? Type of hair. Type of hair is what? A character. Straight hair, curly hair, wavy hair. That is what? A trait. What is the definition of a trait? Variance of a character. Write down a trait. It's a heritable variant of a character. Only two words you have to remember. Heritable variant. What do you mean by a variation? Differences. So a character, say it's eye color. The character is eye color. What are the traits or the variations? Brown, blue, black, green in gray, eye color. So those are traits. Simple, right? Then there's another definition. Third one, phenotype and genotype. Third and fourth, phenotype, right? Highlighting the resource as well as writing them down. Phenotype and genotype. Write them down. What is a phenotype? The definition is in the resource. Observable traits, right? Observable variations of a character. Write down. Phenotype is observable traits of an organism. Write down observable traits of an organism. There are some traits that are hidden. No? Some traits that are hidden, that you cannot observe in the outside of the animal. So I have given to you in the tute, diagram tute, right? I'll refer to this color one as the diagram tute. Huh? It's the diagram tute. If we come to the second page, you will see there's a sort of on the right side middle one, there's a diagram showing the difference between phenotype and genotype. Can you see? Purple, purple, purple and white. Can you see? Online people, can you see? All of you have got the tute, right? Yes. Can you see? What is it? It's the flower color, no? Flower color is purple, purple, purple. And then there's white. So the flower color that you can see is the phenotype, observable trait. Got it? Then who is the counterpart of this phenotype? Genotype. What is the definition of genotype? Write down. Definition of genotype, write down. The genetic makeup. Genetic makeup. Genetic makeup, or we can say set of alleles. We'll talk about this word allele. Genetic makeup or set of alleles. This is observable traits. Can you see the set of alleles from your naked eye? No. Right? You cannot see. Forget about this word allele. Think about this one. Genetics you can't see from your outside. You have to take the DNA out and you have to do DNA sequencing. Right? A lot of complicated techniques you have to do to see the genetic basis. What you can see is the phenotype. So you can see, we'll learn about this, right? Even though the observable trait is purple, the genotype can be different. No? Can you see? Can you see? There's capital P, then capital P, simple P. Both can make a purple color, right? Both can be purple color, but the genotype is different. So what they make on the outside is purple, but inside genetically they are different, right? So always phenotype and genotype is not the same. Just because you can see something from the outside doesn't mean inside they are similar. Got it? Phenotype and genotype. Next definition, genes. Very, very important definition, gene. Right? Very pay close attention to these basic definitions, huh? Right? There's no time for you to sort of I again and again try to understand, right? Just taggala metanim finish the understanding part. Definition, write down. Basic unit by which genetic information is passed from parent to offspring. right? Basic unit by which genetic information is passed from parent to offspring. That is gene. What is another way that you can define a gene? We'll come to that after the next definition. Then the next definition is locus. Singular locus. Mokada plural එක? Loci, right? LOCI, loci. So what is the locus, plural loci? If you take our chromosome, what is a chromosome? It's our condensed DNA, no? DNA ටික ලස්සනට මෙහෙම, it's like a ball of string in the nucleus, chromatin. It's like a ball of string in the nucleus. When the cell is about to divide, it becomes a nicely packed structure that is called a chromosome, no? Yes. So this is DNA. A DNA forms a gene, no? A gene is composed of DNA. You can say that it's composed of DNA. So the chromosome is also composed of DNA, right? We usually study genetics as chromosomes. It's in the nicely packed way, right? So the gene is part of this massive DNA molecule, part of it. So when the DNA is in the form of a chromosome, you can still identify. Say tracker එකක් දාගේ, even when the person is hiding in this chromosome, when the gene is hiding in this chromosome, you can still pinpoint. And when you have found that gene, you can say anna menna metana, the gene is here. Think this is the gene for eye color gene. You have found. අපි සාමාන්‍යයෙන් කියනවා මේ chromosome එකේ මේකට කියනවා P arm එක කියලා. Right? Arms දෙකක් තියෙනවා. We call this a Q arm. Right? Then we can subdivide. මේවා මෙහෙම පොඩි sections වල අර නිකන් අපි තියෙන්නේ නගර සභාව ඇතුලේ ග්‍රාම නිලධාරී මේවා තියෙනවා එහෙම තියෙන, right? ඒ වගේ you can divide the chromosome into small regions, right? And this is the gene now. The locus is what? It's like the address of the gene. Chromosome එක කියන්නේ if it's Sri Lanka or the country, in that country where is the gene living? That is the locus, right? That is the locus. Online people, can you hear me? So write down locus. Definition: a fixed position on a chromosome. A fixed position on a chromosome. Locus is a fixed position on a chromosome. We can write an additional definition for the gene. You can say it's a nucleotide sequence of DNA residing at a specific locus on a particular chromosome. You can now write it down. Another way of defining a gene. It's a nucleotide sequence of DNA. It's a nucleotide sequence of DNA residing on a specific locus. And contributes to the development of one or more traits. Contributes to the development of one or more traits by coding by coding for specific proteins by coding for specific proteins by coding for specific proteins. That is sort of a too extensive definition, right? Araka thama proper. You can say gene is the basic unit by which genetic information is passed from parent to offspring. It's fine, right? Online people, connection issue. Is it laggy? No, right? Right. Then we come to another definition that is actually coming under gene, right? But it's fine. I'll write it still here, right? Definition number 7, allele. Allele, right? Now that's a very, very important definition. Nah? Alternative versions of genes are called alleles. Write down. Alternative versions of genes are called alleles. Alternative versions of genes. Very, very important. Okay. So what do you mean by alternative versions? Why are we having alternative versions? So there's a gene. Alleles කියන්නේ එයාගෙම එක එක versions. So think this is the gene coding for the eye color. Alright? If you take us, an animal like us, how do we call us? We are diploid animals. No? We have two parents. So from one parent we got a set of chromosomes. From the other parent we got another set of chromosomes. Right? We call them mother maternal set and father paternal set. No? So we got two sets. Therefore, if we pair up all our chromosomes, we have a similar one that we got from the father and the mother, right? They are matching, right? There are two chromosomes in all of us that are similar, but one came from your father, but one came from your mother. What do we call those? Any idea? We call those homologous chromosomes, right? So think this is the mother's chromosome, the chromosome we got from the mother, and this is the one we got from the father. Okay, blue one. They are roughly same size, same shape, same length, right? Roughly. Okay. This is paternal, father. This one is maternal, mother. Alright. So there's gene coding for the eye color here. Because we are diploid, we have two parents and we have two sets of DNA, right? So how do we write our diploid? I mean our ploidy level අපි ලියන්නේ 2n කියලා නේද? You know this? 2n නේද? We are 2n. So because we are diploid, we always have two versions of the same gene. We have two versions of the same gene. That means what? We usually have two alleles of a particular gene. Allele කියන්නේ genes තමයි. But they are different versions of the same gene. So if there is an eye color gene here, there has to be an eye color gene on the other chromosome as well at the same place, right? At the same place. So this is also eye color, right? So the gene is what? The gene determining eye color. The different alleles can be different. So for example, your mother gives you brown eyes, father gives you black eyes, maybe. So the gene is what? Determining eye color, but different alleles, different versions of the same gene. No? Then write down underneath allele some points. Huh? Write down beneath allele. Online people, I will send you a photo of the note. Okay, you are writing properly, no? Eh, First point: alleles reside on the same locus. Alleles reside on the same locus on the homologous chromosomes. Online people, purple, orange color, you can't see the orange pen net. What did I say? Alleles reside on the same locus of the two homologous chromosomes. Eh? Two homologous chromosomes. They vary in their sequence of nucleotides. 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 Okay. Then we have to talk about some diversity of alleles, different ways that alleles can differ, right? So the small subtopic, so alleles කියන එක subtopic එකක් විදිහට consider කරත් okay, right? So under alleles we have to study some more topics, some more definitions. Under alleles we have our eighth definition, that is homozygous. Homozygous. Right? Write down the definition of homozygous. Write down the condition of having the condition of having the condition of having two identical alleles. Two identical alleles for a given gene. For a given gene. So imagine, okay, wrote down, right? The condition of having two identical alleles for a given gene. So imagine in both of these homologous chromosomes, mother and father, if they were both black, both parents are giving you black eyes. Black eye කියන එක ගහලා නෙවෙයි, right? Black color eyes, right? That means what? It's a homozygous state. Okay? Both alleles are the same. Can be either. දෙකම brown වෙන්නත් පුළුවන්. දෙකම black වෙන්නත් පුළුවන්. Whatever. Okay? Homozygous. එයාගේම අනිත් counterpart එක මොකද්ද? Heterozygous. Write down heterozygous. Having two different alleles. Having two different alleles. Having two different alleles for a given gene. Having two different alleles for a given gene. Abhi make a, if we sort of, if you like, you can draw this two chromosomes, and for homozygous, black. Heterozygous, you can say black and brown. Okay. The alleles are identical. Right? In fact, you can okay. Simple idea, no? Samana and homo, different na hetero. Very basic, simple idea, right? Okay. Moving on. So we have covered a lot of definitions, no? A lot of important fundamental definitions. So then we have to say, coming back to a little bit of this genotype and phenotype, right? Usually the phenotype is determined by the genotype. No? Usually your genetics determine your phenotype. Usually. But there are other things that is determining your observable traits. That means your phenotype. What are the video and audio is lagging? Still? Yeah, there's a power cut in this area. So generator is running, no? Yeah. Power cut in this area, so there's a chance that internet speeds are dropping, no? You should. Alright. Since morning, if yeah, I think since morning power is not there, no? උදේ ඉඳන් නැහැ නේ. Maybe a small towers වල battery ඉවර ඇති දැන්. They are running on backup power or something. I think maybe 6 or 7 hours they might be out of power. So signal could be dropping. If you have any issue, let me know. Okay. Right. So as I was saying, usually the observable characters are determined by the genetic setup. But what are the other things that can change the phenotype other than genes? This is something additional. Are you entirely, I mean, are you looking like yourself entirely because of your genes? No. From the day you were born, you were fed different types of food, maybe based on the financial situation and also the knowledge of nutrition, right? එක එක කෑම දෙනවා ළමයි. And also some children grow up with a lot of physical exercise. Some children grow up with obesity, maybe malnutrition, ඒ වගේ nutrition problems තියෙනවා, right? And also some children are exposed to a lot of sunlight and තොර skin එක කළු වෙනවා, ඒ වගේ ඔය. Some children are just kept inside, ගෙදර තිය, right? So their appearance and their build, their muscles, so they are maybe having injuries or whatever they look like now is determined a lot, yes, by genes, also by what? Environment, right? Environment. So we can write a small something additional. Your phenotype is sort of equal to genotype plus environment. Neither. Agreed, right? So that's the next statement that it says, right? You can write this down if you like. Phenotype is brought about by the interaction between genotype of the individual and its environment. Your observable traits are a mix of your genes and environment. Okay. So that is how usually a phenotype is determined. Okay. Then next definition that is coming under heterozygous, right? So heterozygous, think of it as a subtopic. Then under that we have two things to talk about, that is called the dominant allele and the recessive. Dominant කියන වචනයෙන්ම you can understand he is the sort of like නිකන් in control one or the controlling one or the major one. Like recessive means what? නිකන් negligible, powerless allele. What is the definition of dominant allele? Very, very important. Huh? Very, very precise definition. Write down. Dominant allele. Precise definition. Write down. At heterozygous state, the allele at heterozygous state, the allele which determines the organism's phenotype. The allele which determines the organism's phenotype by masking the other allele. By masking the expression, masking the expression of the other allele. By masking the expression of the other allele. That is dominant allele. These are fundamental definitions. That's why you cannot master genetics. So, right? Then underneath dominant allele, write down another, let's say, sub something under dominant allele, right? You have to say dominant trait. Dominant trait then you can you can understand the trait produced by the dominant allele is the dominant trait. The trait produced by the dominant allele is the dominant trait. Can you write down the matching definition for the recessive allele. Okay, I'll tell you. The definition of the recessive allele, write down. At heterozygous state, write down, at heterozygous state, at heterozygous state, at heterozygous state, the allele which does not produce, the allele which does not produce at heterozygous state, the allele which does not produce any noticeable effect, noticeable effect on the phenotype, any noticeable effect on the phenotype is the recessive allele. එහෙම ලියන්න ඕනේ නැහැ ඉතින්, right? Then what is recessive trait? The trait hidden in the recessive allele. You have to say that. The trait hidden in the recessive allele. Recessive trait is the trait hidden in the recessive allele. Right. Next definition. Let's take something that was related to Mendel. Mendel used කරපු එකක් මේ eye color, and this eye color I took it just to be more related. It's like human character එකක් ඉඳලා I took eye color, but Mendel couldn't experiment with humans, huh? Right? Because many reasons. Why? If you wanted to observe a human's characters, at least 10 months pregnant බලන් ඉන්න ඕනේ, and sometimes you have to wait until the child is an adult to get the next generation, no? But plants, I think every week or something, no? There are some characteristics of this pea plant, right? Every week you can breed, right? So Mendel can do experiments. Okay, that's why I took eye class more human feature in, right? So in the color tube, so the picture tube, second page, this top diagram, I have given to you the different characters that were, say, used for Mendel's experiments. Different characters that are used for Mendel experiment. What are the different characters? Yes. What are the different characters? Flower color, seed color, seed shape, pod color, pod shape, flower position, stem length, right? Those are the different characters that were used by Mendel. Okay. Those were chosen by Mendel because why? Several reasons. One of the reasons is that you can clearly see the difference. For example, if you take this flower color, ඕනම Montessori ළමයෙක්ට පේනවනේ how to differentiate between purple and white. Very clear difference. Then if you take seed color, yellow and green, you can taggala see, right? And if you say stem length, tall and dwarf, anyone can quickly observe. So he chose those characters for his is. එයාගේ ලේසියට තමයි එයා ඒවා choose කරේ. Right? So those are characters. Stem length, seed color, flower color, those are characters. What are the traits? Purple, white, tall stem, short stem. Those are the traits, no? Different variations. So that's what we are going to talk about next. Next definition, write down definition number main 110. Na? Contrasting traits. Contrasting traits. Write down the definition of contrasting traits. Distinct. Hold on, there was a proper definition somewhere. Contrasting traits definition. You can say distinct contrasting phenotypic forms. Definition of contrasting traits, because I have to give you the resource book one, right? Contrasting traits, distinct contrasting phenotypic forms. You can by observing from outside, you can say these are two different, these two are different, tall or short, purple or white. Okay, contrasting traits. Next definition, pure breeding. Definition number 11. Right, I forgot to mention. So if you take these two chromosomes, think this is purple. What is it? Purple allele. Purple allele. This is the white allele. Okay. So we usually P. So if this is purple and capital P, we don't say W. We usually use simple P. Yeah? Capital P and simple P. Those are the different versions. So white is represented by simple P. So this capital P is dominant. Okay. So when you get capital P, what do you get? Purple. Obviously. Also, when you take capital P, simple P also, what do you get? Purple. Because why? It's the dominant allele. These are simple things, right? Simple P na white. Because why? They are both recessive. Ek dominant ek kari hitiyot, recessive ekena will be masked. Okay? That's the logic. Right. So I was saying pure breeding, no? Pure breeding. Write down the definition of pure breeding. Sorry, not pure breeding. Sorry, pure line, na? Pure line. Sorry, pure line, na? Pure line. Write down pure line. Uniform lines. Uniform lines produced from self-fertilization. Produced from self-fertilization. Produced from self-fertilization. Produced from self-fertilization. Produced from self-fertilization of pure breeding varieties. Of pure breeding varieties over many generations. Over many generations is a pure line. So pure line is what? It's a if you take pure, if you want to make a pure line in an animal, it's a bit cruel, no? You have to do what? You have to cross close relatives, right? Close relatives, fathers and daughters, brothers and sisters. You have to cross very close relatives. Because why? In a plant, you can self fertilize. Pollen in a some certain plants, but you can't do that in animals. So, if you want to create a pure line, what do you have to do? Something we call as inbreeding, right? Close relatives, very close relatives. Relatives, very first relatives, you have cross. Okay, pure lines. Now, if you want, you can write down pure breeding. I mean, not necessary. Pure breeding, the definition is organisms, or you can say organisms obtained by self-pollinating over many generations. That is pure breeding. Organisms obtained by, you can say, plants obtained by self-pollinating over many generations. If you take animals, it's inbreeding, crossing between close relatives over many generations. You can take pure breeding lines. Next definition, definition number 12. We are running out of space, right? Definition number 12, hybridization. Mendel did a lot of hybridization. It's not right. Mendel did a lot of hybridization in his experiments. What is hybridization? Write down. Write down. Mating or crossing. Some animals are mating, plants are crossing. Mating or crossing of two pure breeding varieties. Mating or crossing of two pure breeding varieties with contrasting traits. Mating or crossing of two pure breeding varieties with contrasting traits is called hybridization. What are we going to do then? So these are nice contrasting traits, no? Purple flowers and white flowers. So Mendel, what he did was, එයාගේ greenhouse එකක් තියෙනවා. Mendel has a greenhouse. He has many greenhouses because he's obsessed with pea plants, right? He was always in his greenhouse. We don't know, right? Right. In his greenhouse, he has these what pure lines. What are the two pure lines that you need to do this? He needs to have a purple pure line, a white pure line. How did he obtain those pure lines? He only allowed purple to cross with purple. Okay, he only allowed purple to cross with purple. He only allowed white to cross with white. That's how we got a pure line. Whenever he is trying to do an experiment, he is going to cross this purple and white. Hybridization. Okay. I think with that we are done. No, there are a few definitions, right? Few more. Okay. Hybridization done. Right. Under hybridization, we have some more definitions. First one, monohybrid. Make underneath hybridization, write down monohybrid. Write down the definition. Before that, I will explain. Right. I'll erase this send. You don't need to. You have the basic idea now, right? So remember this example, purple and white example. I'm going to use it, the curtain, right? Because it's a simple example. So Mendel in his greenhouse, he has what? He has pure breeding purple and pure breeding white. What is he going to do? Megam, I'm going to give you understanding of how to draw this process. So the first generation or parents, P, right? Parental generation. What are we going to do? We are going to cross purple with white. So parents lage, what is the genotype? Pure breeding purple, capital P, crossing with pure breeding white. That means what? Simple P, simple. Okay. So I'll write down. This one is purple, pure breeding. This one is white. Confusion never, right? The purple, right? Arakat purple in liver, right? So no problem. No. What do we get when you cross these two people? What do you get when you cross these? There's another next section is the gametes. Again, what allele? So they have two alleles. Both alleles are capital P, both alleles are simple P, simple. So when they are producing gametes, doesn't matter who's male, female. Api gametes kiyala thamai gaye, right? So when they are making gametes, what is the only gamete they can produce? Having capital P, no? Capital P තියෙන එකකින් හදන්න පුළුවන් වෙන එකක් නෑ. Simple P නෑ. මෙයාට හදන්න පුළුවන් simple P තියෙන කෙනෙක් විතරයි. Right? I'm teaching you how to draw this process mega, right? So what is the percentage of capital P? 100%. 100%. Right? Then I'll erase this as well, right? We get the first generation. We call it first filial generation. F1 right? First generation. What is the makeup of the first generation? Right? May again capital P, may again simple P. What is the makeup? Yes. Online people, what is the makeup of this F1 generation? Or the first filial generation? Yes. Sleeping? No. Capital P, simple P. No issue. මෙන්න මෙයාට තමයි අපි කියන්නේ monohybrid. He's a monohybrid. Okay. Then write down the definition of monohybrid. Write down. An organism that is heterozygous, with respect, an organism that is heterozygous with respect to a single gene of interest, resulting from a cross between parents having homozygous condition. Homozygous condition for different alleles of that gene. Read again. An organism that is heterozygous with respect to a single gene of interest resulting from a cross between parents having homozygous condition for different alleles of that gene is called what? Monohybrid. Can you see? These are the parents, homozygous, capital P, simple P, simple P. This is the offspring, capital P, simple P. Heterozygous, monohybrid, simple. Right. Next definition. Before moving on to the next definition, I think have we are complete all clear up till now, right? Any questions online, people? So monohybrid, okay. Next definition, monohybrid cross. Then we are going to complete it. Huh? So we have monohybrid. Now we are going to do experiments with these monohybrids. Huh? Monohybrid cross. Write down the definition of monohybrid cross. Write down the definition of monohybrid cross. Breeding experiment. Breeding experiment conducted between two organisms. Breeding experiment conducted between two organisms. Breeding experiment conducted between two organisms with heterozygous condition. With heterozygous condition. With heterozygous condition for a specific character. With heterozygous condition for a specific character. We are going to do an experiment, right? What is the experiment? We are going to cross. So tell me. So this is their genotype. No, monohybrid ke genotype ka capital P, simple P. What is his color? Purple. Right? What is it? Purple color is his phenotype. No, phenotype. Now what we are going to do is we are going to cross F1 with F1. F1 එකේ ඉන්න අනිත් අයත් එක්ක we are going to cross monohybrids we are going to cross, right? Because we got a lot of monohybrids. Mendel එයාගේ greenhouse එකට ගිහිල්ලා he took a lot of purple ones, he took a lot of white ones and he crossed. Then he got a lot of purple generation, like a new generation of purples. What is he going to do is then මේගොල්ලන් අමතක කළා he is going to cross all these monohybrids, right? That is what we call as a monohybrid cross. So how do we draw a monohybrid cross? It's by something called as a Punnett square. Punnett මේ ඕල කාලේ තැන්ද නේද, right? Punnett squares, simple things, right? This side එකේ could the sperm, this side එකේ could the egg, right? The sperm and this could be the egg. Doesn't matter what side එකද කියලා, right? So what are the possible sperms? What are possible sperms? What are the possible sperms? මේ monohybrid හදන්න පුළුවන්. What are the possible sperms? He can make capital P, no? This is capital P. He can also make simple P, no? What is the percentage? He is having half. So he can make 50% capital P's and 50%. Imagine this monohybrid is making 1,000 sperms. 1,000. There's a chance that 500 of them, those sperms are capital P. No? It can be 495, 480 and whatever, 620 and A again. But roughly 50-50 chance, no? Because why? He's having capital P simple. Same thing, because why? We are crossing monohybrids and monohybrids. Capital P and simple P. What are the results? You have capital P, capital P, capital P, simple P, capital P, simple P, simple P, simple. Right? Then you can say, who are the purples here? Taggala phenotypes. My audio and video is lagging. Others, do you feel it? Anyone else? Senu Kaputa, can you hear it? Any lag? This is purple. This is purple. This is purple. Yeah, white. Neither. Simple P, simple P. Then tell me the ratios. What are the ratios? What are the genotype ratios? What are the different genotypes you can observe? You can have capital P, capital P, capital P, simple P, simple P, simple P. Right? Those are the possibilities. 1:2, 2:1. Right? දෙකක් තියෙනවනේ capital P, simple. What are the phenotypic ratios? Phenotype. What are the two phenotypes? Purple to white. What are the right? Purple to white. 3:1. 3:1. Got it? Different phenotypes. The phenotypes ratio is different to the genotype ratio. Okay. So underneath the monohybrid cross, can you quickly take this down? Quickly, yeah? Online people? Paper class, definitely starting next week. I'm still deciding on the date. The discussion date is the problem. Paper writing, if you can come to Nugegoda and you can write, I'm going to arrange paper writing slots. Right? Probably Sunday after our revision class, I'm going to put a paper writing slot. Okay? 2 hour paper writing slots. You can come and write, and I can mark and give you. And the discussions, I'm still deciding on the date. Right? There's a high chance that the discussions will be purely online, happening from my home. I have a smart board, so I can do it from there. Paper writing slots. One is permanent, that is Sunday about from 4:30 to 6:37. වගේ වෙනකන් you can stay and write. Okay. Physical papers I can mark and give. Online MCQs I can mark. But depends, right? Essays also I can depending on if I have assistants available, right? Yes, but I can try to mark your essays. So that's the situation. Definitely next week I'm starting the paper class. Okay. Okay. Quickly take this monohybrid cross. Quickly take this down. Simple stuff. Pay close attention to this genotype and phenotype ratios. Huh? Going too fast? Anyone who wants me to slow down? Okay. Nil. Right? So using this experiment as the reference, Mendel produced, or let's say presented his first law. Mendel's first law. Okay. By using the results, analyzing the results of this monohybrid cross, Mendel presented his first law. What is Mendel's first law? Yes, online people? You can call it Mendel's first law of inheritance, the law of segregation. Law of segregation. Write down topic. Mendel's first law of inheritance. Mendel's first law of inheritance. Mendel's first law of inheritance. Write down. First law. Law of segregation. Let's quickly cover the theory and then we can do the questions. Huh? We can take a part and we can jump to the question tutor. Write down. Under Mendel's first law, there are two, you can say two postulates, sorry, two principles. Write down. Each heritable character, each heritable character, write down, each heritable character is determined by two heritable factors. Each heritable character is determined by two heritable factors. What are these heritable factors? They are called alleles. Alleles. That's the first law. Or the first part. Sorry, not the first law, first part. Huh? Second part of the law, write down. During formation of gametes, write down. During formation of gametes, the heritable factors or within brackets alleles, during formation of gametes, heritable factors within brackets alleles get separated. What did I say? During gamete formation, the heritable factors within brackets alleles for a heritable character, add the word heritable characters. If I say again, during formation of gametes, heritable factors within brackets alleles for a heritable character. For a heritable character are separated. Are separated. Are separated. Heritable factors are separated. Don't know whether first again. What did he say? Every heritable character is determined by two heritable factors. Those are called alleles. When the gametes are being formed, those heritable factors and are separated. That is Mendel's first law. Very simple, right? Genetics scale, there's nothing to be worried about. Okay. So how do we relate it to this Punnett square? Because you can see Mendel took two pure breeding generations. Pure breeding purple and pure breeding white is the example I took, right? So he crossed two and he got only purple. Then he crossed the purples with purples, right? Then he got a genotype with 1:2:1 ratio. And he got a phenotype of 3:1 ratio. That happened because in the monohybrids, there are two alleles. They only have two alleles because we are considering only one character. For one character, how many alleles do you have? Two. So, the only two alleles found in this monohybrid got separated and got into the gametes, and those gametes got fertilized, they got crossed and produced our F2 generation. What's right? This is a monohybrid cross. This is Mendel's first law. Okay, nothing to stress. Clear about monohybrids and monohybrids crosses. Okay, then we are going to move on to something called test cross. Okay, test cross. If you want to follow the resource book, where do we have the test crosses? Yes. Come to page number 98. The test cross. Page number 98. So where are you? You drew this and did you write down the two laws? Mendel's first law the parts of the two. Right? What was the definition number we stopped? Definition number we stopped 12, 13, 12. Now 3, no? Right. Definition number 13, test cross. Test cross is the next topic that we are going to talk about. Right now, deliberate breeding process performed to determine deliberate breeding process performed to determine deliberate breeding process performed to determine unknown genotypes. Deliberate breeding process performed to determine unknown genotypes. It's, as the name says, it's a test. It's a test one. And we are doing a testing experiment. Okay. Why are we doing this test cross? What is the reason? To identify, as the name, as we just said, unknown genotypes. I think why do we need to determine these unknown genotypes? By observing from outside, you can say. Is it the case? This is the best example you can take. Just because this plant looks purple on the outside, can you say its genotype? Can you assume? Because why? Capital P can be purple. Capital P, simple P can be also purple. So by looking at it from outside, by looking at the phenotype only, you cannot say exactly the genotype, right? The genotype can change. So why are we doing test crosses? To determine unknown genotypes. Okay. So under test cross, write down subtopic monohybrid test cross. Monohybrid test cross. Okay. So there's a law. How do we, let's say there's a method how we do test crosses, right? How do we do test crosses? There is the standard way of doing a test cross is there's an organism. On organism කෙනෙක් ඉන්නවා, we don't know the genotype. We are to give X scale. We are going to cross him with someone who's recessive homozygous for the character we are testing. We are always crossing him with recessive homozygous someone. That is the law. No, not law. The method. We are crossing unknown with recessive homozygous. That's how we do it. So if you take my example, there is a purple flower plant. There is a purple flower. So what are the possibilities for the purple flower color? It can be capital P, or it can be capital P, simple P. No? Or they come in. We don't know which one. So we are going to find out by doing what? Test cross. So what are we going to cross it with? With simple P. Because why? If someone is simple P, he is going to be what? White. He's white, no? But this one is purple, no? Already purple. Now white is purple. So this is the F1 generation. Online people, can you see this edge? This edge you can see, right? F1 generation. We are going to cross our unknown someone with a recessive homozygous to test what is the genotype. Two possibilities. What are the two possibilities? What are the two possible crosses? Let's think. His genotype was capital P, capital P. We are crossing with simple P, simple P. What do you add? Capital P, right? Big capital P, capital P. We are crossing with simple P, simple P. That is the first possibility. No? What are the gametes? What are the possible gametes? G. What are the possible gametes? මෙයාට හදන්න පුළුවන් what gamete? Only capital P 100%. No? Because why? He is only having capital P. මෙයාට හදන්න පුළුවන් මොනවද? Simple P 100%. So when you cross them, I'm sorry, this is not F1. Nah? This is parent. Parent, sorry. This parent, this is F1. So what is the result? You are getting capital P, simple P. So purple, 100% purple. F1 generation එක 100% purple එනවා in this first combination. What is the other combination? I'll erase this. Online people, this is okay, right? This monohybrid cross kata, you know, no? We are now in the monohybrid test process. Okay. අපි test කරන්නේ මොකක්ද බලන්න. What are we testing for? If this unknown is he dominant homozygous එහෙම නැත්නම් heterozygous බලන්න. මොකද එළියට එන්නේ ආව purple. Right? What we can see is he is purple. But why is he purple? To determine his genotype, we are doing the test cross. So what is the second type of test cross we can do? So this is the first test cross. What is the second test cross we can do? Between capital P and simple P and simple P and simple. No. So these are the parents. V. මේගොල්ලන් ඕනේ අපි cross කරන්නේ. What are their gametes? This guy can make what? What are the two types of gametes he can make? He can make capital P half, 50%. And he can make simple P 50%. This one can make simple P 100%. එයාට වෙන option එකක් නැහැ. එයාට PP simple PP විතරයිනේ. Then we are going to cross. මෙයා y cross කරනවා. මෙයා y cross. ඒ දෙකනේ options දෙක, right? නේද? So what are the children? Capital P, simple P, simple P, simple. Ne? So what are the colors? මෙයාගෙන් එනවා purple, මෙයාගෙන් එනවා white. එයාගෙන් එනවා මොකද? White. What is the percentage? Purple to white percentage? 50%. Right? 50% purple, 50% white. නේද? Clear? This is monohybrid test cross. Monohybrid test cross. Mm, write down this example. Right? Write a sentence under monohybrid test cross. Write a small sentence under monohybrid test cross. Write down. Involves crossing of an organism. Involves crossing of an organism. Involves crossing of an organism. Involves crossing of an organism having unknown genotype. Involves crossing of an organism having unknown genotype. X එක having unknown genotype for a selected phenotype. Crossing of an organism having unknown genotype for a selected phenotype. What is our selected phenotype? Flower color, purple, no? Selected phenotype with another organism, from the same species, with another organism of the same species, having homozygous condition, sorry, homozygous recessive condition, having homozygous recessive condition for the same character, for the same character. I'll tell again. It involves crossing of an organism having unknown genotype for a selected phenotype with another organism of the same species having homozygous recessive condition. We don't know if the purple color organism is capital P or capital P simple P. We don't know, because why? Both can produce purple color. We are going to cross him with a white one. The first possibility, if he is capital P, we are going to get all purple. But if he is capital P simple P, we are going to get 50% purple, we are going to get 50% white. Draw these two. Any question? Complete from her. Always think about the possibilities. What are the two possibilities? If someone is purple, he can be dominant homozygous purple or heterozygous purple. So to find that out, we are doing test cross. All right. Shall we take a break? Small one. Online people. Any questions? Okay, the question. If the heterozygous plant only produce one or two offspring, there is a possibility. There is a possibility of all purple, so then may be hard to find heterozygous or homozygous. Ah, yes. This is a very legit question. Yeah, the question is, if a plant produces only one or two offspring, there's a possibility of all of them being purple. What is the fundamental rule of probability rows? ඕනම දෙයක probability, the error of probability law. All these are what? 50%, these 100%, 100% gametes, 100%, 50% gametes. All of these are accurate only with what condition? Very, very, very large sample size. Okay? If this organism is producing only 2 gametes, then dekama වෙන්න පුළුවන් capital P. Right? We don't know. But if he is producing 1,000, there's a chance that it's close to 500, 500. Maybe it can be 480 and 620, 450, 650. එහෙම වෙන්න පුළුවන්. 600, 400 වගේ වෙන්න පුළුවන්. The most fundamental law of probability, genetics is probability laws, is what? The larger the sample size, the more accurate your predictions. You can star mark at the if you're going to forget, right? Larger the sample size, closer your predicted values, right? If you come to resource, there's a nice example of this. Come to page number. It's a very good point that you raised, right? Come to page number 91. That's a monohybrid cross, the one we erased. There's numbers there. Can you see in the F2 generation, 705 purple flowers, 225 white color. So what was our phenotype ratio? Purple to white ratio එක මොකක්ද ආයේ monohybrid cross වල? 3:1 no? Purple 3කට white එකක් ආවනේ. 705 and 224. What is the ratio? Roughly 3:1. Roughly. Well, what is 225 multiplied by 3? 224 no? 224 multiplied by 3? 672. Harinang, it has to be 672 to 224 no? But the experimental value is 705 available. But close enough. No? Mea, if he, so he, on average, he has crossed about 930 වගේනේ cross කරලා තියෙන්නේ නේද? If he cross 5,000, it's going to be very, very close. If he cross 6,000, it's going to be higher, right? So if he cross 6,000, sorry, if he got 6,000, let's say F2 generation, he's going to have what? What are the ratios? 8,000 ගන්න, right? So ඒතකොට 6,000 purple and 2,000 whites එනවා. Very close. Bigger the number of organisms, bigger the sample size, the closer you are going to be to your predicted values. එකකින් දෙකකින් you can't do genetic experiments, right? Mendel එක තමයි මම කිව්වේ Mendelට greenhouse පොරෝලා green pea plants තිබ්බා. He did thousands of crosses. Nathuwa you can't find out anything in genetics because it's all about probability. If you flip a coin 5 times, the chance of having heads and tails could be මොකද එක දිගටම heads හම් වෙන්න පුළුවන් 5 සැරයක්. But if you flip the coin 1,000 times, the probability of getting heads and tails 50-50 increases. 10 සැරයක් උඩ දැම්මොත් it could be close to 50. But if you only flip it maybe 5 times a passerema, sometimes it can be either heads or tails, right? When the number of the sample size increases, the probability laws become more accurate. That's the golden rule lah. Okay, take a 10-minute break. We'll come back at 10:15 nah? Sorry, 3:15. Any questions you can send to the chat and Okay, right. Can you hear, online people? You can hear me, right? Okay, nice. Any questions? No. Monohybrid test process done, no? Right. So with monohybrid crossing, we came to test process. Huh? So monohybrid crossing done, monohybrid test crossing done. So with that, Mendel's first law also done. How many laws does Mendel give? Two. So we are going to go to the Mendel's second law. With that, we have another type of crossing that we call as dihybrid cross. Alright. So that's the sort of the program I have organized, right? We are going to do the dihybrid crossing. Then with that, we are going to complete Mendel's second law, right? It's very organized, unlike your resource book. Huh? The resource book genetic chapter is perhaps, I mean, it's very sort of unorganized, right? Other lessons are okay usually, particularly if you take a chemical basis. Okay, it's a good lesson. Most of the animal form is good, right? But these genetics and molecular lessons are very, I mean, unorganized, right? They are good for someone maybe like an undergraduate credit okay, good, but for A level, it's very difficult to sort of learn by yourself. That's why I started this revision with these difficult parts, right? Online people, you can hear me, right? Any connection issue? No. Okay. Dihybrid process. So we have to jump back and forth in the resource book for this note that we are making. Write down the topic. I mean, sort of a next definition. Dihybrid cross. I'll start here. Nathang I'll run out of space. Right. Definition number 14 වගේ නේද? Dihybrid cross. Write down definition of dihybrid cross. Breeding experiment conducted between two organisms with heterozygous condition for two specific characters. Breeding experiment conducted between two organisms with heterozygous conditions for two specific characters. Dihybrid. It's the sort of adapted definition. Ne? අපි monohybrid එකට මොකක්ද ලිව්වේ? Heterozygous condition for a specific character. එකයි. For two specific characters. Right. So let's go through the resource book example. I mean, let's use the resource book example. Did we write down a definition for dihybrid? No, ne? No, right? We missed that, no? You can write down as a subtopic, right? What is a dihybrid? Before moving on to dihybrid cross, we have to know what is a dihybrid. No? You know what is dihybrid. I want you to have the definition. Okay? What is the gist of it? Dihybrid can මොකද්ද? Contrasting traits for two characters. Right? Isella purple and white, only the flower color. We are now going to follow two characters at the same time. Write down the definition of dihybrid. Huh? I forgot to give you that. Page number 93, you have dihybrid crosses. Write down what is a dihybrid. An organism having contrasting traits. An organism having contrasting traits for two specific characters. Okay, right. Now we are going to follow two characters. What are the two characters we have chosen? The seed color and seed shape. Can you see in that list of this garden pea characters? Seed color, what are the two options for seed color? Yellow and green. What are the two options for seed shape? Round or wrinkled, no? So we are going to follow those characters both at the same time. But that's what we are going to do. Okay. Can you see? If you are just interested, uh, they have given in that same diagram. They have given you the Mendel's exact numbers. Can you see the flower color 705 to 224? So the ratio is 3.15 to 5. Sorry, 1. Can you see? The ratio is 3.15 to 1. It's not exactly 3 to 1. But if you go down to the online, can you see? Seed color to that seed color ratio 6022 to 2001. And can you see? It's very close. 3.01 to 1. No? That means what? When the sample size increases, you are getting closer and closer to the predicted value. So that's the biggest experiment he has done, no? 6000 ගණක් අරගෙන නේද? Total of 8000 plants අරගෙන he has done the experiment. So in that experiment he is very close to the predicted values, no? 3 to 1 එකට very close. And if you look at this pod color කියන එක, can you see? Green and yellow. Online people check the diagram too. Pod color කියන එක, can you see? Green and yellow. එයා කරලා තියෙන්නේ 428 and 152 නේද? එතකොට මොකද වෙලා තියෙන්නේ? 2.82 to 1 වගේ ඇවිල්ලා තින්නේ. එතකොට මොකද වෙලා තියෙන්නේ? The ratios are ටිකක් off, no? That because why is that? The sample size is not big enough. නේද? Something additional. Okay. Dihybrid process. We are going to cross yellow round and green wrinkled. තමයි අපි cross කරන්න යන්නේ. Okay. So who are our parents? We are going to, what are the things we are following? Seed color and seed shape. So yellow is dominant. Huh? So capital Y is yellow. Round means round is dominant, no? Yes, round is dominant. Capital R. Yellow round. Yeah, yellow round. We are going to cross him with pure breeding green wrinkled. So are we writing simple G? No. Is the recessive part of this yellow, no? So simple Y wrinkled na simple R. Genetics. So this is green wrinkle. This is what? Our PO lines. We haven't started the cross yet. pure lines. What are their gametes? මේගොල්ලන්ට හදන්න පුළුවන් gametes මොනවාද? Now be careful, because why? We have two characters we are following. So for each character, how many alleles we have? Two. Capital Y දෙකක් තියෙනවා. That's for the seed color character. So here all together how many alleles? 4. For a gamete that we are making, what do we need to do? We need to give half of all the alleles. So if you are human, අපි green pea plant එක අමතක කරන අපේ ගන්න. How many chromosomes do we have? 46. If you are a male, for every sperm you are making, how many should you give? 23 chromosomes you have to give. So imagine this whatever pea plant has only 4 chromosomes, 4 alleles. He has to give 2 to every gamete he makes. Neither. That is fair. So we are going to give one of each. එතකොට දෙකයි. මෙයාට හදන්න පුළුවන් එකම possible combination එක මොකද්ද? capital Y එකක් දෙන්න පුළුවන්. That's all he has. capital Y R එකක් දෙන්න පුළුවන්. 100%. There's no other option. මෙයාට හදන්න පුළුවන් මොකද්ද? simple Y and simple R 100. Those are the gametes. Okay. Then we are going to cross these two. අපි දෙන්න මේ we are crossing no මේ the sign indicates crossing. So first filial generation, what is the result? We are getting what? A dihybrid with capital Y, simple R, capital R, simple R. Tell me his phenotype. What are the things he is exhibiting? What is his color? Yellow. What is his shape? Round. Because why? Dominant, no? Dominant, dominant. මේ දෙන්න mask කළා තියෙන්නේ. මෙයා yellow, round, 100%. Monohybrid කෙනෙක්ව we have done. Now what we are going to do is we are going to cross these monohybrids. Monohybrid cross. ඉස්සෙල්ලා මතකනේ dihybrids. We are going to cross these dihybrids. We are going to cross these dihybrids and see what's going to happen. This is an experiment. Before I'll tell you the result, let's come up with a hypothesis. What are the things that can happen? Imagine we don't know any genetics. We are Mendel. We are doing this experiment for the first time. Okay. What are the two things that can happen? So I'll come to here. Monohybrid cross එකට යනකො. So we are doing the di again, right? So we are doing the dihybrid cross here, right? So what are we going to do? We are going to cross F1 with F1. Na? We are going to cross with F1 with F1. How do we do that? Capital Y is simple Y, capital R simple R crossed with capital Y simple Y, capital R simple. මේකනේ කරන්නේ. So there are two possibilities. This is possibility number 1. Here we'll draw the possibility number 2. Possibility number 1 is to do this. I'll draw you the chromosome as well, right? So the මේ දෙන්නයි chromosomes තියෙන්නේ කොහොමද? If we draw these chromosomes, there are 2 chromosomes, right? Chromosomes දෙකක් තියෙනවානේ. That's why they are getting separated, right? අපි මෙයාගේ chromosomes ඇන්දොත් මෙයාගේ right? Okay, there's a capital Y here. There's a capital R here. there's a capital Y here, there's a capital R here. එහෙම නේ තියෙන්නේ. නේද? So these two are getting separated and making this kind of gametes. දැන් අපි මෙයාගේ යන්දොත් online people, you can see this small chromosomes I'm drawing, right? අපි මෙයාගේ යන්දොත් මෙයායි කොහොමද තියෙන්නේ? සාමාන්‍යයෙන් මෙතන simple y, මෙතන simple r, මෙතන simple y, මෙතන simple r. කොහොමද තියෙන්නේ? Right? First assumption that we are going to make is may denna inseparable. You can't separate these two people. Simple y, simple r hari, this chromosomes that are on the same, sorry, these alleles that are on the same chromosome, you can't separate. Kila assume karu. Right? So that means what? This capital Y and capital R are a package. Can you see? They are inseparable. No. So similarly, simple Y and simple R are a package. You can't separate. Let's assume. Okay, that is one of the possibilities. Then let's do the experiment and see what's the result that we are getting. So F1 and F1 we are going to cross. Okay. Then අපි එහෙම හිතුවොත් අපිට එන්න gametes මොනවාද? What are the gametes that we can get here? You cannot separate capital Y and capital R නම් මොනවාද the gametes? You have capital YR එකක් හම්බ වෙනවා, simple YR එකක් හම්බ වෙනවා. You get මෙයාගෙනුත් capital YR එකක් හම්බ වෙනවා, you get simple YR එකක් හම්බ වෙනවා. 50%, 50%, 50%, 50%. Okay. Then let's cross these. We are crossing those. මෙයාව මෙයාට දෙනවා. Then මෙයාව මෙයාට cross කරන්න පුළුවන් නේද? Then මෙයාව මෙයාට cross කරන්න පුළුවන්. එහෙමනේ? So what are the possibilities? Y, capital Y, capital Y, capital R, capital R. මේ දෙන්න done. මේ දෙන්න capital Y, capital R, simple Y, simple R. No, අපි එහෙම ලියන්නේ. Right? Capital Y, simple Y, capital R, simple R. Sorry. Right? මේ දෙන්න. Then මේ දෙන්න. නේද? මේ දෙන්නව දැම්මා. There has to be 4 combinations, no? මේ දෙන්නව දැම්මා. Then මේ දෙන්නව දැම්මා. මෙයාවයි මෙයාවයි දැම්මේ නෑනේ. මෙයාවයි මෙයාවයි දැම්මේ නෑ. ඊළඟට මෙයාවයි මෙයාවයි දැම්මේ නෑනේ. Okay නේද? Confused? No, right? හරි නේද හතර දෙනා? මෙයයි මෙයයි. පස්සේ මෙයයි මෙයයි. පස්සේ මේ දෙන්නයි. Okay නේ? හරි නේද? Simple Y is capital R, R, and then ඒ වගේම එක්කෙනෙක් ඉන්නවා capital Y, simple Y, capital R, simple R. ඊළඟට both simple Y, simple R, simple. නේද? Okay නේ? So what are the phenotypes that we are getting? මෙයා මොනවාද? මෙයා yellow, round, මෙයාත් yellow, round, මෙයාත් yellow, round. Only him මෙයා විතරයි. Sorry, green and wrinkle, no? Yes. This is one of the assumptions. This is one of the possibilities. Right? We are going to do the experiment. We are just thinking here. No? This could happen. But Mendel did this experiment. No? Mendel did this experiment and he found out this is not the ratio. Right? So according to this, what is the yellow round to ratio? 3:1 ratio එකක් එන්න ඕනේ නේ yellow round to green wrinkle 3:1 ratio එකක් නෙන්න ඕනේ එහෙම එකක් නෙවෙයි ආ right so come to page number 94 page number 94 okay can you read statement number 1 sorry paragraph number 1 the two characters could be transmitted from parents to offspring as a package highlight the word package the dominant capital Y and capital R alleles or the recessive simple Y and simple R alleles are passed together generation after generation. This is called dependent assortment. This is wrong. මෙහෙම වෙන්නේ නැහැ. Right? But it's one of the possibilities. We have thought about it. අපි ගැන හිතුවා. Right? මෙන්ඩලුත් හිතුවා. Mendel හිතුවා විතරක් නෙවෙයි. Mendel did the experiment and proved that this is not going to happen. Right? According to this hypothesis, only two types of gametes are possible. Only two types of gametes are possible. Thus, the phenotypic ratio of the F2 generation would be similar to that of a monohybrid cross. 3:1 එකක් ලැබිලානේ. But is it a monohybrid cross? We are doing a dihybrid cross. This is wrong. Right? Write down the topic. We have written dihybrid crosses, right? Write down Mendel's two hypotheses. Write down Mendel's two hypotheses. Mendel's two alternative hypotheses for dihybrid cross. Mendel's two alternative hypotheses for dihybrid cross. Mendel's two alternative hypotheses for dihybrid cross. Put number 1, first hypothesis. The two characters could be transmitted from parents to offspring. The two characters could be transmitted from parents to offspring as a package. Next one. The dominant capital Y and capital R. The dominant capital Y and capital R and recessive simple Y and simple R are transmitted together. Sorry, I passed together. Are passed together. This is known as dependent assortment. This is known as dependent assortment. This is known as dependent assortment. Write this down. Name according. If you like, you can draw this. How on the two chromosomes you have the two alleles, and you can show that they are passing as a package. එහෙම වෙන්නේ නැහැ. We'll understand why it's not happening like that with the correct experiment. This is just the hypothesis. No? So this is the dependent assortment experiment, which is wrong. So can you see the two packages? Gametes, capital Y and capital R is a package. Simple Y and simple R is a package. Right? Even package in right? In proper gamete production, there are no such packages. We'll see why. Okay. This is dihybrid cross if there is dependent assortment. Pay close attention to this 3:1 ratio, phenotypic ratio. It's close to monohybrid cross. This is a dihybrid cross. That means this package story is wrong. That's what we are going to see. When the Mendel actually did this experiment, he got a different result. Then next topic, next point, I sent a photo to the group. Yes, I sent a photo. Number 2, second hypothesis. Write down. The two characters are transmitted from parents to offspring independent of each other. The two characters are transmitted from parents to offspring independent of each other. Independent of each other. Therefore, it is called what? Independent assortment. Independent assortment. Independent assortment what is actually happening, not dependent assortment. So we can rewrite this here. That means what? In the proper F1 cross, what's going to happen is we are crossing two dihybrids, capital Y simple Y, capital R simple R with capital Y simple Y, capital R simple R cross. The gametes are alleles are not going as packages. They can cross with anyone. Capital Y can go with simple R, simple Y can go with capital R. And they can mix and pull. Why the mechanism of this mixing we'll talk about later. How do they mix? Second lesson cell division. Meiosis වල මොකක්ද වෙන්නේ? Crossing over. මතකද? Crossing over මතකද? Go and read meiosis. Meiosis වල prophase 1 වල there's something called crossing over. What is going to happen is some of these two chromosomes can break and exchange and rejoin. ඒතර මේ blue එකෙන් කැල්ලක් black එකට යනවා, black එකෙන් කැල්ලක් මෙයාට යනවා. එහෙම වෙනකොට මොකද වෙන්නේ? The capital Y can join with capital simple R. The simple Y can join with capital R. Meiosis prophase 1. Okay. Because of this crossing over, we are getting this chop කරපු හොඳට mix කරපු chromosomes හම්බෙනවා. So what are the possible gametes in this scenario? මෙයාට හදන්න පුළුවන් possible gametes මොනවාද? There are 4 gametes he can make, right? I'll use a different color so නැත්තම් මේකත් එක confuse වෙනවා. He can make මේ දෙන්නව දාලා හදන්න පුළුවන්. Capital Y, capital R හදන්න පුළුවන්. Yes. මේ normal විදිහට හදන්න පුළුවන්. Capital Y එක simple R දාන්නත් පුළුවන්. Simple Y එක capital R දාන්නත් පුළුවන්. Simple Y එක simple R දාන්නත් පුළුවන්. Got it? මෙයාටත් පුළුවන් same. Capital Y එක capital R, capital Y එක simple R, simple Y එක capital R, simple R, simple Y, simple. මේ ඔක්කොම හදන්න පුළුවන්. Only 2 types, 4 types. මේ ඔක්කොමලා 25% possibility. 25% percentage everyone. Then the crossing is not so simple like this, right? 4 times 4 කොටු 16ක පනත් ස්කෝයර් එකක් එනවා, right? මේ side එකට 4 මේ side එකට 4. Okay, that square is given in the resource book. නේද? Page number 94. I'm not going to draw it here, right? What I want you to do is, so we wrote down the second hypothesis now. මේ part එක ලියන්න. Go home and draw the Punnett square for the F2 generation with all the combinations. මේ side එකේ මේ ටික දාන්න sperm. මේ side එකේ මේ ටික දාන්න eggs, right? Gametes. Then fill all the possibilities. You are going to get a phenotypic ratio of 9 is to 3 is to 3 is to 1. You are going to get not only two yellow round and green wrinkle විතරක් නෙවෙයි, you are going to get yes, you are going to get yellow round. You are also going to get yellow wrinkle. You are going to get green round, and you are also going to get green wrinkle. See the diversity increase. ඉස්සෙල්ලා තිබ්බේ yellow round green wrinkle විතරයි. Now there's a in between. There's a yellow wrinkle and a green round as well. Mendel observed this, right? So take this first part down. Keep space for the big Punnett square, right? 16 by 16 එක අඳින්න ගෙදර ගිහිල්ලා. ගෙදර ඉන්න අය online ළමයි දැම්ම අඳින්නේ, right? Right? Start drawing 16 by 16 Punnett square. තනියෙන් අඳින්න, then refer the resource book and check if you have drawn correctly. No. Dependent assortment does not happen. We thought it's happening. Mendel did the experiment and he disproved this. We know there's a chance of it not happening because why? We have studied meiosis. We know of something called crossing over. Crossing over වුණාම මොකද වෙන්නේ? These homologous chromosomes break and exchange. So today homework, go home and study meiosis. Meiosis YouTube video එකක් හරි බලන්න. Okay, I'll send you some YouTube shorts showing crossing over. ඔයගලන්ට වීඩියෝ බලන්න කිව්වට බලන්නේ නැහැ නේ, shorts විතරනේ බලන්නේ. Attention span එක අඩුයි. Right, less than 30 seconds නම් විතරයි you are watching.
One commences with a diploid cell. Its chromatin contains two sets of chromosomes, one inherited from each parent. During this phase, the DNA in the chromatin replicates and condense.
Okay, so with this knowledge, Mendel presented his second law. What is Mendel's second law? It is called law of independent assortment. Mendel, I'll write it here. Independent assortment. Right. So there's only one, main part. Alleles separate and pair up independently during formation of gametes. Right. So it doesn't matter the alleles are located on the same chromosome. When they undergo meiosis, crossing over, they will be exchanged, crossed over, crisscross vela. There will be this much of a diversity of gametes, right? This much of a diversity of gametes. Okay, that is Mendel's second law. Write down Mendel's second law. Independent assortment. Underneath that, write down alleles separate and pair up independently during formation of gametes. Alleles separate and pair up independently during formation of gamete. That is independent assortment. Yeah, yetting there's another clarifying statement. Therefore, two or more genes assort independently irrespective of each other. Underneath that, write down two or more genes assort independently. Two or more genes assort independently irrespective of each other. On the cover of the tube, there's also a diagram that will help you sort of, it's just the basics. We have come a lot further, right? How these genes are located on the maternal and paternal chromosome, or basic story if it's in the cover, right? Basics, right? You are done with the basics, right? Genetics එකේ cover එකේ තියෙනවා, right? Then underneath that, write down. However, this is a very, very important statement. Write down. However, this condition, this condition is valid. However, this condition is valid only in two circumstances. Valid only in two circumstances. However, this condition is valid only in two circumstances. Now, this is a bit tricky, right? There are some situations where these kind of four gametes might not be produced, right? There are only a few two circumstances where we get this 25%, 25%, 25% percentages only in two circumstances, right? So that is because of modern knowledge. වෙලාවට මෙන්ඩල්ට එහෙම circumstances හම්බුන්නේ නැහැ because of the pea plant. Mendel was very lucky, otherwise he would have been very, very confused, right? So one of the conditions is the genes are located far apart on the same chromosome. It's like this. Black and red, no? Yeah. මේව හිතන්න. There's this these are the two chromosomes I'm taking, okay? Imagine those are the two chromosomes, right? Other one has to be blue, no? So what are we supposed to do? We are supposed to cross over, no? Crossing over නේ වෙන්නේ. Imagine this capital Y is here and capital R is here, very close, and this simple Y is here and simple R is here. Now the problem is, what is going to happen in crossing over? Crossing over මොකද වෙන්නේ? A part of the chromosome breaks and exchange. මෙයාගේ මේ piece එක මෙහෙට එනවා. But now, because these two alleles are too close to each other, මොකද වෙන්නේ? When they are getting crossed over, දෙන්නම එකට යනවා. Right? So what is the result? Let's see what is the result after crossing over. So usually what should happen? This black chromosome should have a blue part that is containing simple Y and simple R. And this blue one should contain a bit of black. crossed over now. But because these are too close to each other, what's going to happen is may simple Y and simple R දෙන්නම ඇවිල්ලා. දෙන්න separate වෙලා නැහැ. මේ පැත්තට capital Y and capital R දෙන්නම. Why? Because they are too close to each other. Because they are too close to each other. I'll get it later. Right? For this to properly happen, what should be there? They should be nicely far away like this. මේ වගේ. They should be nicely far away. Right? So if do you want me to draw it? Right? This is the proper way that you should, the two alleles must be, if you want to have that many combinations. What is it like? Ma'am. Capital Y is sort of far away and capital R is far away. Simple Y is in one. Arms then what can happen? Then maybe this R can exchange it, this R. Now okay, no? So what is going to happen? In the resultant chromosomes, we have the nice mixture because of the crossing over. මෙතන ඉන්නවා capital Y, මෙතන ඉන්නවා simple R, මෙතන ඉන්නවා simple Y, මෙතන ඉන්නවා capital R. මේගොල්ලෝ, these two are මේගොල්ලෝ too close. This is too close. This is okay. Right? You can take this down. Right? So only under two circumstances. Right? What is the first circumstance? The genes must be located far apart on the same chromosome. The genes must be located far apart on the same chromosome. Okay, the genes must be located far apart on the same chromosome. That is the first circumstance. Huh? If those circumstances are not there, Mendel's second law is not going to work. They must be located far apart on the same chromosome. Then write down, if not, they escape crossing over. If not, they escape crossing over. If not, they escape crossing over. Draw these two. හරියට පෙන්වන්න that they are too close to each other, and when they are nicely far away from each other, they can properly get crossed over. Got it, right? If they are too close to each other, they act as a package, right? If they are too close to each other, they become this sort of thing. Dependent assortment. It should be nicely far away from each other to for what? For the crossing over to happen. What is the second condition? What is the second condition? මේක තාම ලියනවා. Still drawing. Okay. Right. The second condition is what? The two alleles are located on different chromosomes. Then මේ අපි ගත්තේ on the same chromosome you have the Y's and the R's. On the same chromosomes you have the Y's and the R's. මේ ප්‍රශ්න මුකුත් එන්නේ නැහැ if these are located on what? Two different. Like this. I'll use the same colors for chromosomes, right? මේම හතරක් හිටියා නම්, right? There's one, two like this, and another two like this. Okay, and this capital Y is here, and what? The simple Y is here, and capital R is here, and the simple R is somewhere else. හතරක තිබ්බනම් කිසි issue එකක් නැහැ. Then they are going to get assorted this way, right? No issue at all. නේද? If they were located on 4, මේ වගේ එකම එකේ තිබ්බ තියෙන්න ගියොත් තමයි they are going to be problematic, right? So write down the second circumstance. What circumstances? Where Mendel's second law is valid. Circumstances where Mendel's second law is valid. Write down the second circumstance when they are located in different chromosomes. 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 Right? Remember different, right? No problem. No, capital Y can go with capital R. Simple Y can go with simple R. Capital Y can go with simple R. Simple Y can go with capital R. No issue. මේ එකේම ඉන්න හිටියොත් තමයි මේ ඔක්කොම ප්‍රශ්න. Right? එකේම හිටියත් කමක් නැහැ if they are far away. Far away, you know, they can get what? Crossed over. If they can get crossed over means what? Mendel's second law is valid. වෙලාවට මෙන්ඩල් කරපු එක්ස්පෙරිමන්ට් වල maybe yellow round ඒවා maybe they are located far apart or maybe they are located on different chromosomes. මෙන්ඩල්ගේ ලක් එකට he was not confused because why? The pea plant had these conditions, maybe located far apart or maybe on different ones, I don't know, right? කොහොම හරි Mendel got the second law. It is true, right? When these alleles are assorting independently, right? And so that law was given to the world. නැත්තම් ඌ මේක හරියන්නේ කියලා වැඩි අතහැරලා දැම්මම that law would not be there. But luckily the garden pea plant was obeying these two circumstances. Either they were far apart on the same chromosome, the alleles, or located on different chromosomes. Okay. So that is dihybrid process done. What we have to do is related to dihybrids is dihybrid test process. Okay. Dihybrid test process, let's do next week. Okay. Dihybrid test process, let's do next week. So as homework, not homework, before we finish, every day I do something for you to sort of memorize. All right. I'll put it on the board. Something that you should memorize every day before going home. Right, online people. Shall I erase this? Okay, right? Sort of a warm down exercise before going home. Go to animal nutrition. Nutrition in animals. Animals nutrition. Quickly go there. Online people, in your resource books, go to animal nutrition. Minerals, minerals. Go to minerals. Yeah, you can say non-sister chromatids, yes. Sister chromatids katawa, don't get confused. Sister chromatids in what? Sister chromatids happen because of what? DNA replication, double chain, right? Don't be confused with sister chromatids. Nothing happens between sister chromatids, no. No crossing over happens between sister chromatids. Crossing over happens between homologous chromosomes. So don't get confused with sister chromatids anyway. Yes. What are non-sister chromatids? Homologous chromosomes. Yes. Wi-Fi went out. Right. Go to the resource. Minerals. Where do we have? Oh yeah. Page number 177. Can people, can you hear me? There was a power cut, right? So this is something additional I'm doing in the revision class. Take 5 minutes and we are going to study this. Okay, these are minerals, nutrition, and different functions of minerals. I'm going to classify them based on their common functions. Page number 177 in your animal form and function nutrition. Okay, nerve function වලට අපිට ඕනේ මොනවද? Calcium ඕනේ, potassium ඕනේ, sodium and chlorine ඕනේ. Osmotic and water balance වලට මොනවද ඕනේ? Sodium and chlorine. Enzyme cofactor. What are your enzyme cofactors? Magnesium, iron. These are the common, these are the elements that are involved in performing certain functions together. Bones හදන්න මේවා ඕනේ. Acid base වලට මේවා ඕනේ. Muscle functions, nerve functions වලට මේවා ඕනේ. Right? Then there are some special people like amino acids, amino acids වලට කවුද ඕනේ? There are a lot of amino acids containing sulfur. Then thyroid hormones වලට කවුද ඕනේ? You know this. thyroid hormones වලට කවුද ඕනේ? thyroid එක ලොකු වෙන්නේ මොන අඩු වුණාම? iodine. ගොයිට හැදෙනවානේ. iodine for thyroid. Iron වලට you can say something additional like component of hemoglobin. Maintain acid base balance. Calcium you can say something special, clotting of blood. Yes. So can you see? I think this sort of summarization helps you to study otherwise boring part. No, if you like, you can take down. I mean, it's basically what's in the resource. So now take 3 minutes and try to sort of short term memory එකට හරි දාන්න try එකක් දෙන්න. What are the things that you need for acid base balance, for nerve function, osmotic function, osmotic balance? Okay. All right. Just something new I'm trying, right? නැත්නම් මේ ටික මතක හිටින්නේ නැහැ. Online people, all right? I hope at least few of the elements you can remember with their functions, right? So with that, we can finish for the day. If you have any questions, you can ask, all right? Online people, any questions? So the new tudes will be delivered once you enroll or once you settle this month's fees, right? So from tomorrow onwards, the tube delivery will start. So this month it's genetics, and because there's a high demand to do genetics, but on the other side we are also we have completed nutrition. No, nutrition we have a few things to do, but because in the new month I started with genetics revision, and on the other side we are doing animal form nutrition, animal form revision as well. You have got the tube, right? Animal form revision tubes, right? So paper class will be not whole syllabus. We are going to start with only grade 12. We are only going to start with grade 12. Um, then I'll keep adding things. Okay. So not unit wise. Grade 12 questions only from these 2 months for a few months. Then I'll start adding grade 13 contours. Okay. Any questions? Okay. Keep studying. Only 8 months left. All right. Okay. Online people, any questions? All right. Okay. Thank you for joining.